So I put the map up there, and the blue states there represent the IDEA states, which NIH recognizes in a group as the I want to the underrepresented in NIH funding, and I put little marks up there for, and forgive me, I did not look up where exactly in Montana you actually came from, or Idaho or whatever. So if you're looking up there going, wait a minute, nobody lives out there. But just to represent where people are coming from for this year's conference, symposium. And you can see that we have some new folks from Colorado and Texas, and of course Andy comes over from Memphis, which is sort of Tennessee, but we have quite an array of universities and institutions that are represented here, and I'm going to go and just let you guys, if you can, get your mics on. I want you to go around and just take a moment. That's not going to be in this order. We'll just go around the room, but I'll put Andy on the spot since he's an old timer here, and just introduce yourself a little bit and just give just a little blurb about what you do.
So now I'm on, right? Okay. So I'm Andy High. I'm the director of the Center of Proteomics and Metabolomics at St. Jude Children's Hospital, as Dennis said, right across the river in Tennessee. So our center supports all the basic research and clinical research at the hospital. So we do both proteomics and metabolomics, as well as bioinformatics, a little bit of each. And I'll just say I've been coming here for, gosh, I don't know. A while now, and it's always find something very useful to come out of this. There's a lot of, as Alan mentioned, a lot of interactions and friends that I've made, and we've stayed in touch and led to support letters and things like that, as well as lots of good information that can be used. So I look forward to another meeting all you guys that I haven't met before, and another good conference.
I'm Mike Kinter. I came here from the Oklahoma Medical Research Foundation. It's in Oklahoma City. I drive in, so it's about a 6-hour drive in from the west. My lab, as Alan implied, focuses on targeted methods and targeted quantitation of different proteins, and we have a small discovery service really just for my institution, and just in the last couple of years started picking up on the DIA experiment. So
hi, my name is Min-Kyu Kim. I'm from San Antonio, Texas. My lab is studying lots of protein interactions in cancer cells, and also very much interested in DNA damage signaling in terms of phosphorylation, ubiquitination. So I'm doing a lot of PTM signaling using DIA method. So, and then also I recently joined San Antonio, Texas last year, and then I'm also getting more involved in our mass spec core laboratory at UT San Antonio. So I came here to see what other folks are they're running, how they are running their mass spec cores, and how they are managing services for the core users in the institutions. I'm very much looking forward to it. Thank you.
My name is Yanbao Yu, and I came from University of Delaware. I've been running this proteomics, DDA proteomics for over 10 years, and I do sample prep, do the experimental design, and run the mass spec and deliver data, and even write manuscript. And so it'll be very excited to learn DIA. I got my DIA run, just finished my first DIA run last month. So I'll be very excited to learn from this symposium.
Hi, everyone. Alan mentioned me a few minutes ago. I'm Larry Cornett, the director of the Arkansas Embray here. And as he said, I played a small role in kind of getting this thing started way back when. I have to say it's been really gratifying to see this resource get to the level that it's gotten to. And it's, I'm sure it's in some part due to the support that all of you are providing it. it's definitely, I think, an exceedingly impactful core facility. I know quite a few people in the Arkansas Inbray network at the PUIs use the facility. And it's really kind of raised the level of research that's coming out of the Inbray program. Alan invited me to come over just to kind of see how things are going. I can only stay for a few minutes. This is the year that the Arkansas Inbray renewal goes in. So got a lot to do between now and May 22nd. So anyway, just want to welcome all of you. Thanks for coming. Keep doing all the good work you're doing, and I really want to see this resource continue and go on. It's a great thing.
I'm Rick Edmondson, along with Sam McIntosh. I run the day-to-day operations of the core lab here. Sam's downstairs hard at work, so I think he's going to pop in sometime. But we're trying to keep our core running during this meeting, which sometimes is difficult. But I've been doing protein mass spectrometry for a long time. So one of the great beards that been doing this longer than it's been called proteomics. It's still protein or peptide mass spectrometry to me. But like Andy said, I really look forward to this meeting and what we're going to get out of everything. And so we, even though we're going to be the ones talking, we're certainly not trying to come across as we're telling you how to do something. We're tell you how we're doing things and want to get feedback. I mean, it's kind of funny that we're talking about DIA because I was probably the anti-DIA person for, I don't know, most of the last 20 years. But we're all in now. And so the fact that even Mike Kinter is going to talk about DIA, I think solidifies that. So, but welcome to everybody, and glad you're here.
Hi, my name is Noor Tahir, and I work at Dartmouth College up in New Hampshire at the Institute for Biomolecular Targeting, which is right now is mostly a biochemistry core facility. And we're in the early stages of trying to offer mass spectrometry support first to the local research community at the college. My personal background is in biochemistry and structural biology, and I look forward to meeting all of you and knowing you. And thank you to our hosts for having us here, and I look forward to learning a lot. Thank you.
Morning, everyone. My name is Helen Sum. I'm actually from Thermo Fisher. So I know Alan and Rick about a year ago when I first got assigned to this territory to me. So I'm from Houston, Texas, but now cover quite a few states, as a technical sales specialist. So I didn't know this quantitative national proteomics national resources until I met Andy, and since sorry, not Alan, and since then I started to learning this program, and found out this is such a great program. So and this is the first symposium that I attend myself. So I'm so thrilled to meet every one of you, and many of you probably are Thermo customers. And the topic this year is DIA. So this is a big area that Thermo has put in a lot of resources and trying to drive the development of the area. So if you have any questions, feel free about your Thermo products, or if any issues, please come to me. And last, I want to thank Alan and Rick and Dennis for organizing such a great symposium. So thank you so much for having me here to be part of it. So I hope everyone enjoyed this 2 days here, and look forward to speaking to each one of you. Thank you.
Hi, my name is Eduardo Caligari. I am coming from South Dakota. I have been running the proteomic core facility for a long time. This is, I don't know how number 10 I'm coming. It's a great opportunity to be here. Our facility is mainly involved discovery, but we try to do identification, characterization, and certain quantification to level it free and sun level it. The TMT in general, we try to start to establish a relationship with the Arkansas here for the IDEA National. And the reason of coming this time is our goal is try to start to work on DIA. I was doing something which is not exactly the same thing, which was the original MS2DI from the water. It's not exactly the same, but something else. And again, thank you also for here, the guy from Arkansas, for the support every time that I needed something, either way for advice, for protocols, or do some meeting for figure out, and also for invite me to attend.
Yeah, hi. My name is Mikhail Golovko. I go by Misha. I am from North Dakota, just a neighbor, like 500 miles, still a neighbor. Yeah. So we are doing mainly small molecules for 20 years, or metabolomics and some kind of lipidomics, mainly lipidomics, but still have very small instrument QE that we are trying to use for proteomics. And these workshops, I have been here a couple of times, was a great resource to transfer our effort to support proteomics needs of our local community. It was great. We are doing DDA almost all the time for metabolomics, but now we're just looking how we can apply this for proteomics. And yeah, thank you very much. It's a great resource for what we are doing.
Yeah, my mic already died. So I'm Stephanie Barham. I'm the director of the bioinformatics for our national resource here. So I work more on the after it comes off of the instrument part with the analysis, but you'll hear more from me tomorrow. So, pass on.
Yep. Hello, my name is Eric Schultz. I come from the University of Montana, where I've been research faculty since 2018. I specialize in virus host interactions, primarily with cytomegalovirus. And I've used this facility a couple times. I actually have samples ready to send again as soon as I get back. I also am the manager of our mass spectrometry core at the University of Montana, and it's a small core. We have triple quad and a QTOF, and we are sort of a sister core to the Montana State IMBRE core over in Bozeman. But thanks to a group of researchers that were absorbed by the University of Montana, we just were able to purchase an Exploris 480, and we are really looking to bring DIA and all the other things it can do to Montana or the region around. So I am very thankful to be here, and I hope to learn as much as I can about how to make this work for us up there.
For protein mass spec and flow validation. The mass spec that we have is actually exactly, it's not really the latest, but it's working properly. We have it for about 7 years, and we use these methods, and we have about 34 users from the island, north, south, west, universities, and private institutions related to the program and RCMI. We look forward to improve our facility with better implementation and also apply the but something that apply.
Hi, good morning. I'm Horacio Serrano from University of Puerto Rico. I'm director of the Clinical Proteomic Lab. Right now I have a 3 project in Alzheimer, cancer, in thyroid and pancreas. Thank you for the first opportunity.
My name is Kirk Hansen. I'm from the University of Colorado. I've been doing proteomics for about 25 years. I'm here to figure out how to do proteomics. I'm still learning. It's an exciting space, but right now I'm currently trying to integrate a targeted assay with about 800 internal standard peptides into a DIA assay. And a lot of my research is focused around characterizing the extracellular matrix, so polymers of proteins that are insoluble using standard biochemistry methods.
I'm Chris Hemme from the University of Rhode Island. Our state is so small that our 2 stars can't even fit on it. It was a good effort, though. I run our INBRE Molecular Informatics core, which is our data sciences core. So I do a lot of bioinformatics and increasingly more proteomics.
Morning, I'm the times 2 from University of Rhode Island. So I'm the director of a core facility, and we just get involved in the proteomics last year. And in our renew, we proposed that we are going to support our local proteomics users. So Chris and I, we are here, and we try to learn something, and went back, and we will build up our proteomics service. Thank you.
And good morning. My name is Xingui Sun. I'm from an ISU Health Science Center support. I'm a director of mass spec core and the redox core. And from this meeting, we learn out we start from proteomics, and right now we're trying to do more DIA model. That's
Hi, I'm Kavita Sharma, and this is my first time. I got to know this opportunity from one of our co-director, Dr. Ken Cornell. I think he had suggested me to go here, and I'm assistant professor in College of Pharmacy from Idaho State University. So basically, I'm from the south of Idaho, and I have started learning proteomics. We have AB-SciXQ trap, and I use Skyline software just to look for the targeted analysis. But most of my previous experience is in small metabolites. So I support Idaho State University. We have a very small core lab. We don't do outside samples so far. We just do the university sample from chemistry, biology, or chemical engineering to support our own small group. So I just came here to learn more for the core, how to operate core. And also we are planning to apply NSF grant for MRI, which I am thinking of Thermo Fisher because Dr. Ken Cornell got last year. So he suggested me to go with that direction, and that's why I'm here. Thank you so much for giving me this opportunity.
We have a few people that are joining us by Zoom. If you guys could, if you can hear us, give us an introduction, if you would.
I'll start if that's okay.
Start with
Can
you,
hear me,
Aaron.
Okay. Good morning, everyone. My name is Aaron Story. I'm a mass spec lab director. I learned everything I know about mass spec here at the IDEA National Resource for Quantitative Proteomics.